Okay, here we go, guys. This is a Chapter 3 review, protein structure and function. We'll go ahead and start off with the basic structure of proteins. They're made up of monomers known as amino acids. Amino acids have three key parts to their structure. They have an amino group, a carboxyl group, and an alpha carbon that has four bonds. On one end, you have a hydrogen, and on the other end, you're going to have these residual groups, also known as side chains. There are 20 different side chains, so there are 20 different amino acids, and the side chains determines the amino acids reactivity. So here you have nonpolar. That's a review from the previous chapter. Then here you have your polar amino acids, and then you have your aromatics. Aromatics are because they have this ring right here, this hexagon with double bonds inside of it. Then you have your positively charged ions and your negatively charged side groups. Positively charged side groups. So the side chains determine its reactivity, and it's also going to determine the function and structure of the protein. So how do these proteins form? As I mentioned to you before, you have your basic amino acid structure right here. This is your basic amino acid structure. What they do, you get all these amino acids, the different amino acids, and you start putting them together to make a polymer. So you get a bunch of monomers, as you've seen before in a PowerPoint before, and it makes a polymer. This process is called polymerization, and this is done through dehydration synthesis or condensation reactions. They're both the same but different name. And then when you do that, when you have that polymerization process, then you can form all these different structures with proteins. And the key part to forming these proteins is the peptide bond. As you can see here, you get the carboxyl group of one amino acid, the amino group of another amino acid, and you remove water. So water is going to come from these two hydrogens here and this oxygen from this carboxyl group. These two hydrogens are part of the amino group from another amino acid, and this is the oxygen from a carboxyl group of another amino acid. They go through the reaction, water is removed, and it forms this peptide bond. And what's happening here, the nitrogen and the carbon are sharing electrons, which gives it characteristics of a double bond, making it really strong, but yet flexible. All right, but yet flexible. So here's another look at these different peptide bonds. You have a peptide bond here, another peptide bond here, and another peptide bond here. So you have a tripeptide. Like I said earlier, the peptide bonds are very flexible, and also they can rotate in a 360-degree angle. Start forming these polypeptide chains. Here you have another view or another schematic of the process of the polymerization process. You have two amino acids. So you have the carboxyl group right here and the amino group. You remove water and you end up with this peptide bond. Here you have a tripeptide. That means there's three different amino acids. So here you have one side group, the second side group, and the third. There's three characteristics to these polypeptide chains. You're always going to have a beginning, which is going to be the N terminus, which is an amino group, and then you're going to have the end, which is a C terminus, which is the carboxyl group. And then you're going to have all these side chains. The orientation of these side chains is going to determine the function, the reactivity, and the folding and the protein structure. Okay. There's also a numbering system. The beginning, once again, is the N terminus, and that's where you start off with amino acid number 1, and it goes forward in a sequential order until the very end, until you get to the C terminus where you have this carboxyl group. Always remember, another characteristic of polypeptide chains is that they're very flexible and they can rotate as well, since the peptide bond can rotate 360 degrees. These are the three basic structures of proteins, which we'll go over. We have a primary structure, secondary structure, tertiary structure, and a quaternary structure. In the primary structure, it's linear, all right, and it's simple. There's some folding, as you can see here. Here you have amino acid number 1, as we spoke about orientation or direction of the polypeptide chain, and here we have amino acid 129. Notice that none of these amino acids interact with each other. Okay, there's no interaction between the amino acids. So that's why it's nice and simple. It's linear with some folding. But then when you get to secondary structures, now you start having interactions with the functional groups within the polypeptide. So you start seeing some interactions within close proximity amino acids starting to have some type of structure or some type of bending, which gives you the alpha helix, which is like a spiral staircase, or beta plated sheets. Just get a piece of paper, fold it up like an accordion, and that's kind of what it's going to look like. Then you have tertiary structures. Tertiary structures, similar to secondary, however, the interactions occur with residues that are far apart. So here the residues are close to each other. All right, remember residues are the side chains. So these guys are close to each other. They're in close proximity to each other. But over here, they're distant. Say you have amino acid number 3 reacting with amino acid number 75. Okay, and same thing over here. Here again, you'll say you have amino acid number 50 interacting with amino acid 450. Okay, and they're going to have different bondings within these tertiary structures. So the different bondings are hydrogen bonding. This is a good example of it right here. You have hydrophobic interactions. Remember, hydrophobic means it's water fearing. You have some van der Waals interactions. Okay, you have some covalent bonding and some ionic bonding, as you could see here. Now, quaternary structures, very similar to the tertiary structures in the way they fold and so forth, but it's made up of globs. I call it the globular protein, because you get several of these tertiary structures to form your quaternary structures. Okay, your quaternary structures. So you have several polypeptides held together by the same forces found in tertiary structures. So here's hemoglobin. Hemoglobin has 4 different structures. All right, 4 different structures. And this is a perfect definition of them. It's an aggregation of 2 or more peptides. All right, an aggregation of 2 or more peptides. To aggregate means they come together. So that's a quaternary structure. So these structures is critical, the protein structures are critical to its function and what they're going to do in cells and in your body. So protein folding is very critical to its function. So here you have a normal protein, okay? A normal protein, the folding process happens in the beginning of, or right after the protein is formed. But once you denature a protein, it loses its biological activity. Denaturing, there's a lot of denaturing agents out there. One, for example, is heat. Another one is pH, being acidic or basic. So what happens is this protein is functional right now, but once it denatures, all right, it unfolds and it's no longer a functional protein. It's no longer a functional protein. And in some cases, you can renature it and it regains its activity. It's a reactivity again, or its activity again. It's functionality. But there's times when that doesn't happen. And a perfect example would be egg whites. Egg whites, when they're nice and clear right out of the egg, or just nice and clear, but then when you add heat to them, they turn white. You can't renature that protein. It's already been denatured and it's no longer a functional protein. But yet our bodies can still break down those egg whites, and we use them to make more proteins. We break down the amino acids found in the egg whites. And then protein folding is regulated. All right, here you have a ribosome, which is making a protein, and then you have a couple of chaperones. Molecular chaperones are like chaperones you would have on a field trip. They just want to make sure everything is okay. Nothing is going wrong. So what happens is these chaperones guide the process of folding. Once the folding is completed, then the protein is a functional protein. All right, so these functional proteins are very, very diverse in our bodies. There's so many things proteins can do in our body, and it's vital that their folding is correct, so they could take care of what they need to take care of in our cells and in our body. For example, a lot of our body's reactions would not run without enzymes. Enzymes are catalysts. What they do, they speed up chemical reactions. The key thing about enzymes, they have an active site. All right, and they fit like a lock and key system, so they have to fit perfectly to what they call a substrate. All right, so this is the active site. The active site is going to be a lock and key system. Once the substrate attaches to the enzyme speeds up the chemical reaction and then produces the products, and then the enzyme can be reused again. Okay, a good enzyme would be lactase. Lactase breaks down the sugar lactose. All right, and the lactose is found inside our milk. Then we have antibodies. That's why I have up here defense. Antibodies are critical to your immune system. Antibodies are the memory system of your immune system. They always remember certain microbes or certain foreign materials, so when they see it again, they attack it. All right? Next one is movement. movement. All your muscles are made up of proteins. They're made up of two proteins, which is actin and myosin. And these proteins both contract in order for us to be able to make these muscular movements through our bodies. So next time you move, think about these little filaments, these little myosin and actin proteins, all squeezing together and relaxing in order to make these movements that you're doing right now. Then we have proteins that are found on cell membranes. Remember, cell membranes have to communicate with the outside environment and send that message down to the nucleus where it controls all the cell's activities. So what happens is these proteins receive messages from the outside environment, send the messages down to the nucleus, and certain processes occur. Another function of protein is structural proteins. As I told you, your hair, your nails, your skin has protein in it. It's called collagen. So as you get older, this collagen loses its elasticity. So that's why we have droopy skin and wrinkles and so forth. So those are structural proteins. And the last type of proteins I'm going to talk about are transport proteins. There are two types of transport proteins. This type of transport protein found here is part of a passive transport that allows stuff to go in and out of the cell, but it requires no energy. So that's why it's passive. So it takes molecules from high concentration to low concentration down the concentration gradient. On the other hand, you have protein pumps, also transport proteins, but they need energy to move molecules from low concentration to high concentration. So remember, those are transport proteins. Well, this is it for chapter 3. I'll be sending you a link to take a quick quiz over what I just spoke about. Hopefully everything works out, and I hope you enjoyed this because there's many more to come.